Okay. Okay, we're going to waste no time this morning. Good morning, everyone. I just want to thank everyone for hopping on. Joined with me this morning is Joel Williams and Shannon Schumacher, and I will just get right into it and have them introduce themselves, and then they can jump right into the presentation. So, Joel, Shannon.
Hi, I am Shannon Schumacher. I'm an RSA with QLF. I am just here to moderate some questions and ask Joel some questions on his presentation he's going to share with us. I will say I've been lucky enough to go to some of Joel's courses and take some of his online courses, and they're very informational. So I hope you guys enjoy what we're learning as much as I do. And feel free to throw any questions in the Q&A box on bottom, and I will run through some of those at the end. So go ahead, Joel.
Awesome. Okay. Thank you very much, Shannon, and welcome everyone. Great to be here today, and thanks for the invitation to come and speak. Do you want me to introduce myself? Do I need to do that too, or I guess I can quickly, maybe. I'm sure some of you probably, hopefully, know me already, but so I'm an independent plant and soil health educator, originally from Australia, where I worked for many years, and then I moved to the UK, and I've worked around Europe for many years, and 8 years ago I moved to Canada, and I'm based over there now, but still very much work internationally in those 3 locations, particularly in North America, Europe, and Australia. And yeah, I do a lot of education and that kind of thing, and a little bit of consulting as well. I'm dialing in today from Wales, over in the UK, so it's the afternoon here. It's a reasonable hour of the day. So yeah, nice to be with you all, and hope you all enjoy the session today. So let's dive into it. We're going to talk about nitrogen, and particularly around nitrogen processes in the plant, nitrogen use efficiencies, and a little bit of deep dive into some nitrogen metabolism, a little bit on foliar applications as well. So let's get to it. So firstly, there are a range of different processes in which the plant engages with the use of this important element of nitrogen. And the first one, of course, is uptake. First, the plants need to acquire nitrogen from the environment. And of course, there's a lot of nitrogen that comes from the soil, so we have uptake from the soil. But of course, we also have other processes like nitrogen fixation that also deliver that nitrogen into the plant. But when we talk about uptake, there's kind of 2 main categories here. We would say there's some inorganic nitrogen and organic nitrogen forms in which the plant can take up. And we're going to deep dive into those 2 particularly. Once we take up that nitrogen, we need to start to metabolize it, turn it into other compounds, functional, other things that the plant also requires. So a lot of the nitrogen that's taken up from the soil is used to make amino acids, and amino acids are, as often called, the building block of all life. So amino acids then get stitched together to form proteins, and proteins are very important, as you all know, from a quality point of view. We're often interested in high protein from a quality nutritional point of view, but there are many other proteins that also provide more functional purposes. Many of the proteins are enzymes. They catalyze reactions. They help the plant grow in all sorts of different contexts. So protein synthesis is a big outcome of, and a big reason why plants scavenge nitrogen. But there's plenty of other organic molecules that are important here too. Chlorophyll, of course, contains nitrogen, their DNA, their RNA, etc. as well. So we start to metabolize, we turn that nitrogen into other functional compounds, organic compounds. And some of those need to also be moved around or generated in different parts of the plant. So there's a need to move nitrogen around the plant. And it is, of course, one of those highly mobile nutrients. And actually the plant will move nitrogen around. It will metabolize it into amino acids, and it will move nitrogen around, particularly in that amino form. It is also moved in other forms, but predominantly in the amino form. And then we also have this process of remobilization. This is where many of you will be familiar, of course, that the plants will take nitrogen from the leaves, and they will send that to those reproductive organisms, for example, reproductive organs, during that phase. So, to seed fill, to grain fill, that kind of thing. So they will move proteins, break them down into amino acids, translocate those amino acids up to the developing grain, for example, where they'll restitch those together into those proteins. And indeed, just as one small example to highlight how important it is to optimize the nitrogen status in the leaf, and of course the leaf is the solar panel of the plant, it's also where so much of the work happens. Then in terms of finishing plant development, so into reproductive stage, just to illustrate how important it is that during senescence, during maturation, the plants will actually remobilize and strip a huge amount of that nitrogen out of those leaves and send that up into that grain for reproductive purposes. So just as one example there, in cereals, it's suggested here that around about 50 to 90% of that final nitrogen that's in that seed is actually derived from some of these remobilized sources. So these are, in essence, some of the different processes in which the plant makes use of some of these different nitrogen sources and forms. So let's get a little bit into these different forms. Now, plants actually have the ability to take up many different types of nitrogen. And the traditional ones that we've thought about a lot in high production agriculture generally has been some of the inorganic forms like nitrate, like ammonium. And of course, we also use urea as a common source of fertilizer as well. Now, beyond that, the plants can take up organic forms as well. And really, the form of nitrogen in which plants take up actually really depends on what's available in the environment. So, as an example, here you can see here's nitrate. Well, if the plant detects that nitrate in that soil environment, that nitrate present is also sends a signal. It's also a signaling molecule. The plant will detect that, and it will switch on a particular gene that upregulates and codes for one of these nitrate uptake channels. So it will build a special protein in the roots there to absorb that nitrate. So these are can often be highly specific uptake channels that are specific to specific nutrients. So in response to the nitrate being present, the plant will produce these nitrate uptake channels to then absorb. Similarly with ammonium, if it's ammonium present, well, we have different specialized ammonium uptake channels that will take up that ammonium. Similar with urea, we do have an uptake channel specifically for the urea molecule. And then we also have specific uptake channels for some of these organic molecules, such as amino acids. So the point here is just to say that actually the plant responds to the environment. And in traditional agriculture, we have often said that it is the inorganic forms that are the more important ones, the dominant forms, the nitrate, the ammonium, etc. But really, that's primarily because that's what we have applied in agriculture, and that's what often cycles in agricultural soils. If you look to natural soils, into wild native ecosystems, etc., that's where you see plants in those wild systems. They do make use a lot more of amino acids and organic forms because those are what are predominantly available and cycling. So again, they're just responding to that environment, and plants, domesticated plants, are just responding to how we manage them in agro ecosystems versus those natural ecosystems. So the take-home message here is that actually plants are very adaptable, they're very flexible, they can use a range of these different sources. And in agriculture, if we want to optimize nitrogen use, supply, uptake, and use, then it's actually a smart move to try and diversify and use a variety of sources of nitrogen. So this is really just summarizing that point. Here's the traditional view that said, actually, these inorganic forms, these smaller, simple ions, are the predominant source of nitrogen uptake. And we thought that actually that was the only forms of nitrogen that the plant could take up. We used to think that these larger organic molecules, like amino acids, like chains of amino acids called peptides, or larger chains called proteins, we would used to say that, oh, those are too complex. They are, molecularly speaking, they are too large, too complex, and actually the plant cannot take up those large, complex organic molecules. We now know that that is definitely not the case. Indeed, these can indeed provide a highly favorable form of nitrogen for plants. And of course, it extends even beyond that. Even whole bacteria even ginormous things, relatively speaking, to these simple ions. Even whole bacteria can even be engulfed into the root cells. And of course, that's that rhizophagy cycle in which the plants will strip nutrients out of those bacteria. So even these biological forms are also highly favorable and able to be used by plants. And the reason, the core reason why we should be a bit more interested in some of these organic and biological forms is that these are a lot more stable in the soil. When we talk about nitrogen pollution in the environment, nitrogen leaching, nitrogen off-gassing, and the like, it is these inorganic forms that are the highly reactive forms that are more prone to leaking, more prone to those losses. So the benefit of widening our lens on nitrogen nutrition and considering some of these other processes and forms is the stability of them, and that helps us synchronize supply and demand. Because the problem with the inorganic forms is, particularly when we use starter fertilizer, we put a lot of inorganic forms up front, these highly reactive, leaky forms up front, but we have tiny plants, tiny seeds, tiny seedlings that have very low demand. We have not synchronized supply and demand at all. And this is where we open the door to some of these losses. So some of these organic forms, etc., are much more stable and bring that advantage. Now, let's dive a bit into metabolism. No matter what forms of nitrogen the plant then takes up, those forms of nitrogen ultimately plug into what's called the metabolic pathway, or the nitrogen metabolism, in which the plant will use those forms of nitrogen and ultimately turn them into some of these other functional important organic molecules, such as the proteins we mentioned, the chlorophyll, the DNA, and the like. This is really what the plant wants. This is kind of the end game. This is the useful and functional molecules that help plants grow. Nitrate really is just a building block to help the plants get there. Same with urea, same with ammonium. They're all important sources of nitrogen, yes, but at the end of the day, the plant doesn't keep them as nitrate or ammonium. It wants to turn them into these useful and functional organic molecules, such as proteins. So in order to do that, in order to convert, for example, nitrates along this pathway, we need other nutrients. There are other synergistic nutrients that help to catalyze, that help to facilitate this conversion along this stepping stone, along this pathway. So as an example, you can see here nitrate. Actually, first nitrate has to be converted to, I've kind of shortcutted, I've summarized this step here. Actually, nitrate first has to become nitrite, and then nitrite becomes ammonium. And in order to convert those reactions, we need especially molybdenum, really important here, along with sulfur and also iron. So if you don't have enough molybdenum or sulfur or iron, that nitrate gets stuck as nitrate. It cannot move down this pathway. It cannot jump down these stepping stones because we have not got the right nutrients to form part of the enzymes. The enzymes is the thing that catalyzes this reaction, facilitates the reaction. So you can have a buildup or an excess of nitrate, a backlog of nitrate, which of course is not good, particularly in a forage context for livestock. We don't want too much nitrates in their feed, but even also from a human health point of view, we don't like too much nitrates in our feed, in our food, or in our drinking water either. So that's where we need to think beyond just nitrogen management, think about the other nutrients that work with that nitrogen. So let's jump on to the other examples. Here's urea. In order to convert urea, once the plant takes up urea, we need nickel. Nickel is an essential trace mineral that the plants require very small amounts of. Now, it's also a heavy metal, as many of the other trace minerals are. So, we also need a word of caution there to be careful when handling some of these trace minerals, etc., which are also heavy metals. So be mindful of their use in that context. But just to acknowledge, nickel forms part of this urease enzyme, and that's the one that converts urea along this pathway into ammonium. So without nickel, if we have a deficiency or a limitation of nickel, urea can also build up and cause a backlog or a bottleneck of that urea. And that excess urea can be one of the contributing factors in nitrogen scorch, a urea toxicity. So, okay, both those two move along this pathway towards ammonium. Once we get ammonium, now we're going to convert that ammonium into organic forms into amino acids. And the first amino acid in this process is called glutamine. And in order to help synthesize that glutamine, we need manganese and magnesium. So once again, if you have a deficiency or limitation of these two here, that ammonium can also get stuck and it can build up and start to accumulate, causing ammonium toxicity. And that is also one of the other common causes of scorch. Nitrogen scorch can either be an ammonium or urea toxicity. And the underlying root causes of that could be a nickel deficiency, a manganese, or a magnesium deficiency, for example. Okay, now we have some organic nitrogen. When I say organic nitrogen, I mean nitrogen that is attached to carbon. It's got some carbon now attached to it. And we start to then stitch those amino acids together to form some of those larger complex proteins. And there are many, many important nutrients that drive this end process because there are many, many different proteins. And so it depends on which protein is being synthesized as to which nutrient is required to build that enzyme to convert that reaction. So the top three here are particularly important. Phosphorus, sulfur, magnesium are very, very important for good protein synthesis. But also, again, manganese, boron, potassium, zinc, copper, they all additionally also play a role. Okay, so I think the take-home message of this slide is really just to say that when we think about nitrogen management, we have to go beyond just thinking about nitrogen itself. Nitrogen is not an island. It works in conjunction with all of these other nutrients. And therefore we need to consider those, be managing those, monitoring those. I'm not saying you have to be applying those, but I'm saying you do want to know whether the plant has enough of those or not. And of course, therefore, a plant analysis becomes a very useful way. A leaf analysis or a sap analysis becomes a useful way to then assess the status of some of these other nutrients and ensure that we have adequate supplies in order to optimize this process of nitrogen metabolism, be that for a protein synthesis outcome or for other some of these other organic molecules as well, like chlorophyll. So widen the lens, make sure we think about some of the other nutrients. You can see there's a lot of trace minerals on this list. We have moly, we have iron, we have nickel, we have manganese, we have boron, we have zinc, we have copper. So you can see there are many, many trace minerals that are very important for optimizing nitrogen utilization. And that's, I know we all understand the importance of nitrogen, but I'd just like to emphasize that nitrogen does not do what it does without the help and support of some of those other trace minerals. So they also need to become part of the discussion. And then we have this idea of, well, which form of nitrogen is better? And look, they're all a bit different. And they all do slightly different things. And actually a balanced supply of them all is generally a bit of a good idea. However, when we start thinking about some of these organic molecules, they have a bit of a particular benefit if we are supplying amino acids, if we are supplying peptides, some of these organic forms, in that they are closer in this, if I go back here, in this kind of metabolic pathway, these are all closer to the end. So the stepping stone to kind of finish the job is just very small. It's a small jump in the chain, a small link in the chain to finish this, which means these are very efficient forms of nitrogen because they save the plant's energy. If we feed the plant nitrate, there's a lot of stepping stones to get to the end goal. So you've got to step into nitrite, step onto ammonium, step into aminos, and step into proteins. You've got many steps in the chain there. And each step is an energetic drain. It drains energy of the plant and therefore leaving less energy for other growth processes. So in essence, what we are talking about here is shortcutting that metabolic pathway. So if you feed the plant ammonium, as this example, look at all these steps we've got to take. Well, what if I just feed the plant some of these amino acids? We've got less steps to take, and therefore we save energy for other growth processes. So this is the benefit of some of these organic forms. If we can feed the plant more of these aminos and peptides and proteins and deliver these already organic forms to the plant, then indeed we are helping that plant save energy, and that energy that's saved can be used for, you name it, other growth processes. It could be for more tillers. It could be to fill more grain, fill bigger grain, to grow more roots, produce more biomass, to fuel its defense chemicals, you name it. if we're saving energy, we can use that on other growth processes. So these are more efficient, and this process is called shortcutting that metabolic pathway, or metabolic shortcutting. So let's dive into a specific example of this, just to break this down in a little more detail. Here we have a great paper talking about the bonus, the carbon bonus of organic nitrogen forms, which can enhance the nitrogen use efficiency. So I'll just read you this quickly. The chemical form of nitrogen that is taken up, whether inorganic, such as nitrates, or organic, such as amino acids, may significantly influence plant shoot and root growth and nitrogen use efficiency. The key point here is that the carbon cost, or the energetic cost, of assimilating those already organic nitrogen forms and finishing the job into proteins is lower than that of those inorganic forms that have more steps in the chain. And it is particularly because the organic forms are further up the metabolic pathway, but they also contain carbon embedded in them already. And it is that carbon bonus that makes it more beneficial for plants to take up organic versus inorganic nitrogen. So let's do some carbon economics and break this down in detail here. So here's nitrate, here's ammonium, and of course these are inorganic forms of nitrogen, meaning there's no carbon there, so we have no carbon. So in order to convert nitrogen into aminos and onwards into proteins, we need to start stitching carbon into that in order down that metabolic pathway. So there's an energetic cost here. And so the carbon cost of making use of 1 gram of nitrate, so to convert 1 gram of nitrate, we will use up 5 grams of carbon. 5.81 grams of carbon is what this is saying. So we are draining 5.81 grams of carbon to convert 1 gram of nitrate down that metabolic pathway. And of course, nitrate has no carbon embedded in it, so our final net costs remain the same. Similarly with ammonium, here we have it is 4.3 grams of carbon to convert 1 gram of ammonium. So you can see ammonium requires less energy than nitrate to form proteins, etc., to be assimilated. And that is because ammonium is further up the metabolic pathway than nitrate. Nitrate has to be first converted into ammonium. So we bypass that step. We don't need to do that, and therefore we save energy. So the carbon cost is lower to make use of ammonium, and that's why ammonium is a more efficient form than nitrate. Now, the challenge with nitrate is, the challenge with ammonium, is that ammonium has quite a narrower range of toxicity. So it's not fair to say, oh, we could just feed the plant 100% ammonium, because actually if you have too much ammonium, it can start to cause that scorch, that toxicity that I mentioned. But nonetheless, it's just important to understand that ammonium is further up the pathway, therefore we use less energy. Now let's have a look at these two amino acids. This is glutamine and arginine. These are some of the organic forms that are nitrogen and carbon embedded. Now you can see there's still a carbon cost here, and that's to do with the uptake of those, the acquisition of those amino acids. I mentioned at the beginning of the lecture that the plant has to build an amino acid uptake channel to absorb that amino acid from the soil. So there is a carbon cost to acquire nitrogen, to translocate organic nitrogen around where it is required. There is still a carbon cost associated with this. However, the difference is that we are now absorbing organic nitrogen, so we are absorbing embedded carbon. There is carbon embedded in that amino acid. So that carbon, embedded carbon, is like a rebate. We get a bonus, we get a rebate. So that comes off the cost of acquiring and using that nitrogen. So here we have a carbon bonus, embedded carbon, rebate, that comes off the cost, and therefore our net costs are then lower. So the most efficient form, the most energetically efficient form of nitrogen to feed a plant is some of these organic molecules for this very reason. Okay, so that's a nice kind of example, putting that into numbers. Now, when I say we are draining carbon, we have to stitch carbon into the nitrate, stitch carbon into the ammonia. Well, we've already got carbon here. Where does the carbon come from that we are stitching into nitrates? So we are stitching into this ammonium. Where do we get carbon from? Well, of course, it comes from the process of photosynthesis. And that means when the plants are photosynthesizing and we take that carbon dioxide and we stitch it into glucose, the product of photosynthesis, if we are feeding the plant predominantly inorganic nitrogen, that means that more of our photosynthetic carbon, the glucose production, has to be funneled down this pathway in towards stitching carbon into those inorganic forms of nitrogen and making the various amino acids that you can kind of see here. We're not going to go into this chart in detail. I'm just pointing out that the more inorganic nitrogen you feed, the more of your photosynthetic carbon has to come down this pathway and form all of these amino acids. And if you were just feeding these amino acids in the first place, well, that photosynthetic carbon, that glucose, could be going elsewhere into other growth processes. Okay, so now the thing about organic forms of nitrogen is that they also are particularly good at stimulating root development. The inorganic fractions generally encourage a lot more above ground biomass, and that can be good too. Obviously, above ground biomass is what you're harvesting, so there is a balance here. But in terms of the effects of the different forms of nitrogen on the physiology of the plant, the take-home message here is that the organic fractions, the organic forms of nitrogen, they do encourage a lot more rooting and root biomass. And that's what this study here is showing. And of course, more root biomass is really important for resilience. If you want bigger root system, that's going to help you access more soil volume, and that's going to help you access more moisture from that soil for resilience, but also help you access more nutrients from that soil. So what you're looking at here is a neat study on corn, where we are comparing 3 different sources of nitrogen. We are comparing ammonium, that's the black bars here, so inorganic ammonium form. The gray bars here, you can see this is an amino acid form that we're feeding, and the white bars here you can see is a protein hydrolysate. And a protein hydrolysate, of course, is taking a high protein material, digesting it and breaking it down into a mixture of amino acids and peptides and maybe still some undigested proteins there. So it's going to be a bit more of a mixed input, plus typically it's got a few other additional nutrients, essential nutrients that might have come from the original source material. And we're looking at the comparison of these 3 different forms of nitrogen on a whole range of different root traits here. We're looking at root length, root biomass, various root properties. So we're looking at all the various root properties. Now, the key thing about this study is that the amount of nitrogen in these 3 treatments is identical. So pound for pound, this whole discussion, the quantity of nitrogen in each 3 of these treatments is identical. It's the exact same amount of nitrogen, but one in an inorganic form, amino acid form, protein hydrolysate form. But the exact same overall number of elements of nitrogen are supplied equally, but they have very different effects on biomass production because some of them are shortcutting the metabolic pathway, saving energy, and therefore that energy saved can be used for greater root biomass. So you can see this general trend here across the board, across all of these various root traits that they're measuring, that you have this general step up that the organic forms, pound for pound, unit of nitrogen for unit of nitrogen, are inducing a greater biomass response, even though they are supplying the exact same amount of nitrogen. Let's have a look at another study illustrating the exact same thing. This is quite a recent one, just from last year. Here's a neat little study, just a lab-based study here again, but nonetheless illustrating this principle here. Here, we're growing these little plants on these little growing plates. And so what they've done here is they've supplied organic nitrogen, amino acid nitrogen, across the plate, and we are growing these little plants and measuring root hairs. Over here in our second treatment, we're going to use now the inorganic nitrogen. It's the nitrate form that we are then supplying 100% of. And over here in our third treatment, you can see we have split the growing plate in half. On half of the plate, we are supplying organic nitrogen, and on the other half of the plate, we are supplying nitrate, inorganic nitrogen. Now, this is one plant with a split root. So half of the root system is growing in the organic side, half of the root system is growing in the inorganic side, but it is one plant that has its split root. And we look at the impacts of this on our root hairs. And you can see here, here's our first treatment, 100% organic nitrogen. We have a nice, the biggest amount of root hair length that you can see here. And visually, you can see this here. Here's the organic fraction. You can see lots of little root hairs all through there. So we have lots of root hair formation under organic nitrogen supply. Let's compare that to that second treatment, which was nitrates. Here it is over here visually. You can see that the root hair formation has declined significantly. Over here on our split root treatment, on one half of the plant, on half of the root system, you can see that we have, again, much higher root hair production versus the other half of the same plant that has now dropped right off. So clearly the effects are somewhat localized. It wasn't a systemic response that if we supplied amino acids over on this half that they were so well translocated and helped over there. Didn't seem to be that effect. It was quite localized there in terms of the impacts. So again, take home message, organic forms of nitrogen are great for root development. And if you're in a dry land context, that can be very, very important. So here we are looking again at the carbon economics or the energetic cost, the metabolic cost of those two different forms. And what you're seeing here on the left-hand side here is we are looking at the amount of our root mass and the nitrogen uptake. So here we're talking about of the nitrogen that was taken up, be that the nitrate form or the amino form, once nitrogen was taken up, how much biomass can I produce with that amount of nitrogen? And the trend that you're seeing here is, so the red is nitrate and the blue is organic, is the amino acid. So of course, what you're seeing here is that the blue is lower than the red. So we have less nitrogen uptake. We have less nitrogen in the plants from that organic form. But then if we look at the root mass, the amount of biomass we're able to produce with that lower amount of nitrogen, you can see it extends further into the right. So we have more root mass over here from the organic form, but we have less nitrogen in the plant. So we have less, but more. And that's why the blue line is kind of below the red line and extending further to the right. So there's the metabolic efficiency, the metabolic shortcutting. Per unit of nitrogen, we are able to produce more biomass with those organic forms. And just a quick sideline here before we move onwards, just to say that amino acids are also really important for stress tolerance. And you name the stress, particularly the environmental stresses here, you name it, amino acids ultimately, they play a role in all sorts of stress alleviation. And that's because they are able to be stitched together into special proteins that are essentially help the plant deal with that stress, that help to alleviate the symptoms of that stress. So whether that's, heat stress, high UV stress, high temperature stress, heavy metals, for example, salinity, high salt stress, drought stress, waterlogged stress, you name it, pretty much any environmental stress, which, let's be honest, every year we have a range of different environmental stresses. You name it, amino acids will, in one way or another, help the plant to produce the specialized proteins that will alleviate the negatives of that stress. So just reading you a little quote from this paper, metabolic regulation involving amino acids is considered the primary strategy for plant protection and survival under these adverse living conditions. So anything stress-related, aminos are also really important. So let's move on to nitrogen discussions, and we'll link this bit into our foliar. So nitrogen in the soil. The problem with nitrogen applications to the soil is that nitrogen in the soil is very, very leaky. And again, it's particularly the inorganic forms that are most leaky. So we see off-gassing. Some of the ammonium can off-gas. Some of our urea applications can off-gas as ammonia, and that can volatilize off up into the air. Some of the nitrate under waterlog conditions can also go through a process called denitrification, which leads to nitrous oxide production, and those nitrous oxides will also off-gas up into the atmosphere. So we have ammonia gas, we have nitrous oxides gas, which can vapor off into the air, and we also have then, of course, the nitrate leaching. So it is the inorganic forms that are highly, highly prone to these reactions, and these forms of nitrogen are highly leaky, highly reactive, highly leaky, and this is where we have a lot of these nitrogen losses and nitrogen inefficiencies creep in, because nitrogen use efficiency typically hovers around about 50-odd%, give or take in different contexts, but on average about 50-odd% of what you apply is going to enter the plant. So enter stage left, the opportunity for foliar applications here is that when you foliar apply your nitrogen, you're keeping some of that, you're keeping that nitrogen out of the soil. And if you're keeping it out of the soil, that nitrogen is not going to be prone to these reactions and these losses. So if you foliar apply your nitrogen, you bypass the soil, you deliver that directly into the leaf where the plant can take up that nitrogen, preventing some of those losses. And when you prevent losses, you can use less nitrogen because you're losing less. Okay, so if you lose less, you can use less. So what about this foliar discussion? Is it a natural thing? Does it happen in nature? Do plants even use foliar leaves for nutrient acquisition? I thought, why do they have roots? Like, why do plants evolve roots? Of course, look, roots are the primary organ for nutrient acquisition from the soil. So I'm not saying to completely abandon that, but I'm just saying let's acknowledge that foliars can make a valuable contribution, and let's use the tools in the toolbox to their most optimized. Let's get the best out of whatever tools are in the toolbox. So yes, we can improve nitrogen management through the soil. And yes, we have an opportunity to optimize nitrogen management through the leaf. Here's some examples in nature. Kelp, aquatic plants, sea plants. Yes, they are absorbing plenty of minerals from that mineral-rich ocean water into that leaf. Dust storms, believe it or not, can also provide a valuable source of nutrition. Here's a nice visual example of this, but there's a great study looking at applying dust on things like wheat, chickpeas, on arid plants. They also did it to corn, which had the same ability, a bit less than wheat and chickpea, but nonetheless, they demonstrated that plants are able to solubilize nutrients from the dust on the leaf and solubilize those and take those up through the leaf, particularly phosphorus. Meaningful amounts of phosphorus can be absorbed from dust by solubilizing that phosphorus out of the dust in through the leaf. So yes, foliar dust storms also help to feed the plant. Animal waste, insect waste, so you name it, fecal matter, urine, of course, contains urea and other soluble nutrients. Some of that, particularly in you livestock integrated folks, any of your urine coming down onto the leaf is going to be absorbed through that leaf. So yes, animal waste in nature is taken up through the leaf. And lastly, even fog has been demonstrated to contain ammonia gas. And that ammonia gas in that fog can also be breathed in through the stomata and absorbed through the leaf. So I share that just as a quick example to say that, yes, it happens in nature. So that opens up this question of nitrogen use efficiency. And the typical definition of nitrogen use efficiency is nitrogen uptake efficiency. And we define this as the total nitrogen in the plant relative to how much fertilizer we applied. Okay, so of how much did I apply, how much of that did I get into the leaf? And that's an important piece of the discussion, but we can also think about nitrogen in the context of this other definition, which is nitrogen utilization efficiency, defined as how much plant biomass or seed yield can I achieve relative to the total nitrogen in the plant. And so here I'm saying, once we get nitrogen into the plant, how much biomass can we produce? And of course, to answer that question, we need to refer back to our metabolic shortcutting. And it is the organic forms, because they are more energetically efficient, they are able to shortcut that process. We can produce more biomass per unit of nitrogen with the organic forms as compared to the inorganic forms. So how much biomass can I produce per unit of nitrogen? And the answer to that depends on those different forms of nitrogen. And as we've outlined, the organic forms are more efficient in that regard. So what about the mode of action of foliars? What's the argument against foliars? We say, you know what, foliars are all good, but we cannot supply all of the plant requirements through the foliar. Nitrogen, potassium, the demand is too high. We can't apply enough nitrogen or potassium, for example, through the foliar to meet the crop requirements. And this is always the argument used, saying, so therefore foliars are irrelevant. And I would agree with that statement, apart from the end, it does not necessarily make foliar approach irrelevant. It's a misunderstanding. I'm not saying that we are going to apply 100% of the requirements through the foliar. We're trying to supply a percentage, a component. And when we do that, that foliar application actually helps the plant acquire nutrients from the soil. Foliar applications is really all about helping the plant take up nutrients from the soil. And this is because this is the mechanism or the mode of action of foliars here, is that when you foliar apply nutrition and you prime photosynthesis with that foliar applied nutrition, you produce energy. And some of that energy is sent down to the roots to grow more root biomass, to produce bigger roots. And if you produce bigger root system, that root system is going to acquire more, expand, explore more soil volume and acquire more nutrition from the soil. So you're helping the plant obtain more nutrition from the soil because some of that foliar applied nutrients is going to stimulate root biomass growth. And secondly, some of those foliar applied nutrients are going to prime photosynthesis, and that's going to prime root exudate production. And here we have, of course, all of those root exudates, the sugars, the amino acids, the carbohydrates, the lipids, all sorts of other compounds. They are dependent on mineral supply to prime photosynthesis, to prime the production of those. So foliar nutrition is also about priming root exudation. And when we prime root exudates, that concoction of carbon compounds feeds the soil biology. And when we feed the soil biology, they cycle the nutrition from the soil. So foliar application is all about stimulating and helping the plant acquire nutrition from the soil. And the amount of nutrient that the plants will take up from the soil is far greater than the amount that you're foliar applying. So the foliar application is just priming the pump. That's all you're really trying to do. And here's a couple of quick examples of this. Foliar ammonium has been demonstrated to increase the uptake of nitrogen from the soil. Foliar potassium demonstrated to increase potassium uptake from the soil. Foliar urea, enhancing soil nitrogen and phosphorus uptake. Okay, so that's really all we're trying to do with the foliar. We're trying to help the plant. We're trying to apply small targeted amounts directly into the leaf, bypassing the soil to prevent those losses, but nonetheless helping the plant acquire nutrition from the soil. And that's the important mechanism. So that's where foliars can be more efficient. There's a couple of different ways in which this plays out. Some of the factors that drive the efficiency gains of foliars is things like even coverage. In a liquid, you have very nice even application of the nutrients compared to sporadic placement of granules, or down the row where it can be a bit more sporadic and maybe not all plants are requiring some of that nutrition. Whereas in a liquid foliar, we have a perfect even canopy. Each and every plant can be obtain a bit of that nutrition through a liquid foliar application. Once we put nutrients down in the soil, of course, they're more prone to lockups, antagonisms, etc. And some of those losses, the volatilization, the leaching, etc. And of course, sometimes roots do not function optimally if we have drying soil, if we have salty soil. Sometimes the root uptake is not so optimized in those difficult conditions. So even if you have nutrition in the soil, it doesn't mean that it's coming up into the plant. And then the foliar is where we have greater control of what form of nitrogen goes into the plant. Because though if you put nitrogen down into the soil, it will engage in all of those reactions in which will change or convert the nitrogen into different forms. Whereas when we foliar apply it, you can ensure that you get that nitrogen into the plant in the form that you applied it. So for example, if you put urea in the soil, urea doesn't stay as urea. It will convert back into ammonium and or nitrate. If you put amino acids in the soil, those amino acids might get eaten by the biology in the soil. Whereas you put those things in the foliar, you're more likely to get them into the plant as in the form that you want. Okay, I can see I'm running out of time already. I've gone way over. So I'm going to have to really scoot through a couple of these last slides, actually. And in this
You're
last
good,
section,
Joel. You could take your time on these last couple ones.
can I? Okay, thank you. Okay, so let's summarize some of these key points. I'll just make a comment that there are a few other talks on YouTube you'll find where I do go into some of this as well. So if any of this little next section piques your interest, you'll find a bit more information online. Just have a search. Okay, so foliars can be highly variable. Okay, I will acknowledge that. And we have to design for success. And there are important factors to consider in order to design for that success. So I'm just going to summarize some of the key ones here. But as I say, you can refer online for more information. So I break it down into these 4 categories. Firstly, it's the formulation. What goes in the spray tank? That's really important. Secondly is the application, how we apply it. Thirdly is what is the crop or the plant species. And lastly is the environmental conditions during the time of application. So all of these are really, really important for a good success. And I'm just going to cherry pick 1 or 2 of these out here for today's presentation. So one, I would say water quality is a really important factor. If you're using kind of bore water, groundwater, this type of thing, you can often have a lot of contaminants in that water, particularly in more alkaline waters. So if you have high pH water, this can have things like carbonates or bicarbonates. You can also have like high calcium or magnesium. Some waters can be quite salty, high salt levels. Some waters can be quite have high iron levels. So all of these things essentially are contaminants in that water. And that means that when you solubilize foliar nutrients into that water, those contaminants can bind to those foliar nutrients, rendering them locked up or unavailable or lesser available in the spray tank before you've even applied them. And this is also true of our pesticides as well. We see this very much true about pesticides. They can be rendered inactive or partially inactive in the spray tank before we've already applied them because some of these contaminants will bind and lock up. So I would really emphasize, if you can, look at rainwater harvesting and saving, if you can do that, or look into something like a reverse osmosis kit to clean out those contaminants and have pure water. You will see a better response from your foliar nutrition and your pesticide applications. Spray pH, I think, is also an important one. You want to drop that pH down to around about 5 to 5.5 as a general rule. Certainly a maximum of 6. I would say in that kind of 5 to 6 is generally ideal for foliar nutrient uptake. And then I would lastly just also emphasize the importance of including a carbon source or a chelating agent. And that chelating agent, that carbon source, is also going to help to bind to the foliar nutrients, but instead of locking them up, it's going to enhance the uptake. It's going to chelate and improve the uptake. Let's have a couple of quick examples of this. So visually, this is just a nice slide that kind of summarizes and explains this. Rather than apply the nutrient on its own, combine it with a carbon source, which will be like a sponge. It will bind and chelate that nutrient, and actually that's going to enhance the availability and enhance the uptake of that nutrient in a foliar context. Also true of a soil context. If you apply that down into the soil, that's going to reduce some of the lockups and the leaching and the losses down in the soil as well. Here's a nice example of this with a neat study with molasses, combining molasses with some liquid nutrition, nitrogen and phosphorus. And this was a study from Australia done on a very light sandy soil, sugarcane production area, where, of course, in sandy soils, they're highly, highly prone to leaching and losses. So what they did in the study is simply apply some N and P fertilizer, and they compared that to that N and P fertilizer with molasses added on a carbon source. And what they found is that carbon source added helps to stabilize the nutrient and induce a slow release mechanism. I'll quickly read you these quotes. Nutrient immobilization occurs when the application of ready available organic materials to the soil stimulates the growth of soil microbes, enabling them to take up luxury amounts of nitrogen and phosphorus from that soil environment. That immobilized nitrogen and phosphorus, because it's taken up into the bodies of the biology, this is stored within microbial cells and is released or mineralized in the plant available inorganic form at a later date, essentially creating a slow release. Now, again, there's a good example of the previous paradigm. We're talking about all the nutrients have to be mineralized back into those inorganic forms to be taken up. Of course, we now know that that is, yes, true and a part of a mechanism of nutrient supply, but some of these organic molecules can also be taken up. Some of those bacteria themselves, once they gobble up and eat that nitrogen phosphorus, if they enter that rhizophagy cycle, then we're supplying nutrition through that biological form. So they don't have to be mineralized to be released. This is a good example of the molasses stimulating the biology to eat the fertilizer, grow microbial biomass, and that biomass itself can be a source of fertility to the plant through that rhizophagy cycle. So our results suggest that the molasses amended liquid formulation can be considered an enhanced efficiency fertilizer, harnessing the nitrogen and phosphorus immobilizing capacity of soil microbes with microbial sequestration, ensuring proportionally more fertilizer remains in the soil after 17 weeks. So we saw this greater soil storage of the nutrients where we simply just added carbon and encouraged the biology to eat and store the nutrition in their bodies, inducing this longer term slow release mechanism. Okay, and over here we're looking at comparing a quick little nitrate leaching sub-experiment here. So they were applying nitrate through these leaching columns where they were applying straight nitrate versus nitrate with molasses, with carbon, and comparing those two. So adding molasses to the nitrate charging solution at the same concentration as the liquid fertilizer, so 10% by volume, this stimulated the microbes in the soil column to immobilize or to eat, to gobble up some of that nitrate, to immobilize 18% of that nitrate within 2 hours and 95% of that nitrate by the 4th day of incubation. So here I was talking about these highly reactive, inorganic forms of nitrogen that are problematic. Well, you can turn them into positive forms of nitrogen, biological forms, by simply adding some carbon, encouraging the biology to eat, store it in their bodies, prevent lockups, prevent leaching, prevent these losses. It's as simple as that. So the take-home message here is just add carbon. Add a carbon source anytime you're putting fertilizer out. Add a carbon source. It's as simple as that. Here we have a nice example of adding a carbon source to a urea granule in a granular context. So we have two different treatments here. We have straight urea, and then we have adding a zinc sulfate coating to that urea. And then we have a third treatment of adding, with that zinc sulfate, we also add molasses. And so here we're looking at the urea release rate. Here's our control, straight urea. And you can see we have rapid release over time. We have quick and rapid release of that urea. Where we add a zinc sulfate coating, we slow that release a little bit here, and you can see that there in the red. But where we add zinc sulfate plus carbon source plus a bit of molasses, you can see we slow it down again. We have a lower and slower release, and therefore greater stabilization of that nitrogen. So there's a nice example of a way to process a fertilizer granule by adding some of the carbon sources. So we also see this in the soil nitrogen levels. You can see, okay, not huge statistically difference between these two, but between these treatments, but you can see where we have the added molasses and zinc sulfate. It's we have a bit more holding on to nitrogen a bit better in the soil. But over here we see the impacts of this on the microbial biomass. So of course, where you've added the big standout one here, you can see where we have increases in microbial biomass. It's, of course, where you've added the molasses, where you have the highly digestible carbon source, which the microbes can feed on and grow their microbial biomass. And in doing so, they're gobbling up and eating some of those nutrients, storing it in their bodies. So the biggest impact on microbial biomass is where we have digestible carbon, in this example, in the form of molasses. So, of course, then those organisms are going to be storing that carbon. Now, we also have things like humic substances, just as an example, just to provide an alternative here. These have a slightly different mode of action. When we talk about fulvic acid, humic acids, really what you're doing is applying a nice big sponge, and that sponge has lots of charged surfaces, and those charged surfaces will bind to, they will complex and chelate those nutrients, and therefore prevent those lockups, but also enhance the uptake. So this is a bit of a different mode of action. We're more so adding a big sponge with high surface area to stabilize the nutrients. And that's a little bit different from highly digestible carbon sources, which stimulates the biology to eat the fertilizer. And this was a quick example of seaweed. I might scoot over this one just quickly. It was just really to highlight that what is seaweed? Seaweed is mostly carbon. It's carbohydrates, a bit of protein, a few other things. Look at the fertilizing capacity. Look at the mineral nutrition of the seaweed, this yellow bar here. The actual essential macro micro minerals are not really the main thing of what seaweed is. Seaweed is all about some of these other carbon compounds, some of the other plant growth hormones, some of these biostimulant type compounds. It's not the fertilizer per se that we're adding. It's about indirect stimulation. And what they are summarizing here in this review paper is how, after application of seaweed, how that impacts the nutrient status of the plants. And you can see a few examples here in maize, or corn, as you guys like to call it. Seaweed application increased nitrogen, phosphorus, and potassium uptake. Here's in, what's another one maybe relevant for you folks? Wheat here. Seaweed application enhanced grain potassium as an example. There's a couple of examples here from canola or oilseed rape. You can see we're increasing shoot nitrogen and sulfur, for example. Okay, here's soybean down the bottom. Seaweed applications increasing nitrogen, phosphorus, potassium, sulfur in the grain, nitrogen, phosphorus in the straw. Remember, we did not apply any of these minerals. Look at the amount of the fertility. It's insignificant amount of NPK, etc. But yet it had an effect on increasing the nutrient supply in those crops, in those plants, in the plants or the harvested product, even though we didn't apply those minerals or an insignificant amount of those minerals. So again, it's about the carbon source providing an indirect stimulation of the soil biology and the plant root system, stimulating root development, in which we are simply helping the plant acquire more nutrition from the soil, even though we haven't actually applied any to the soil. So chelation, the key idea of chelation here is really just to say that we're going to combine a carbon source or a chelating agent with our fertilizer, and that will bind to that nutrient. And the key benefit of why we do this is to wrap that up with carbon. And the key reason to do that is to neutralize the charge of that element. So by that I mean, of course, cations are positively charged, anions are negatively charged, and a cation or an anion, when we foliar apply it, both of those charges just mean that we have more resistance against the uptake. So if we can neutralize the charge, and that's what chelation is all about, chelated nutrients are neither cation or anion, we bind and we neutralize the charge. That charge neutral molecule can then pass through the leaf more effectively. And that's why many of you would have heard seen many products on the market these days as chelated nutrients, chelated trace minerals, and this is the reason, is that when we chelate them, we do enhance the uptake. And there are many good carbon-based chelating agents like molasses, like humic substances, like kelp seaweed, like amino acids. And of course, urea is a particularly interesting form of nitrogen because it already has some carbon embedded in it. Urea is carbon-based nitrogen. Urea is an organic molecule. Now, when I say organic, of course, I don't mean certified organic, but I mean in terms of its chemistry, it has carbon. So urea is a particularly good form of nitrogen to apply through the foliar. I mean, amino acids are the number one, and they are the fastest to be absorbed through that leaf. But second is urea in terms of speed of uptake, then ammonium, then nitrate. And the reason that urea is particularly good here is that it is carbon-based. It has carbon and it is a charge neutral molecule. That's not a cation. That's not an anion. And it is this charge neutrality of urea that helps it be absorbed particularly efficiently, just like amino acids, through the leaf. So your two prime forms of nitrogen for foliar is aminos and urea. It's not to say that you can't use ammonium or nitrate, but they are not so rapidly absorbed. They are lesser efficient in that regard. And here's a nice visual example of this. On the left-hand side, you're looking at urea uptake through foliar over time on two different grasses. And here's your first interval. Interval here is 5 hours. So you can see after urea application, the bulk of that absorption happens very rapidly, within an hour or two, within a few hours, 2, 3, 4 hours. The bulk of the job is done. You can see how rapid that is. So this is the advantage from a foliar point of view, is speed of uptake. The quicker you can get that nutrition into the leaf, the quicker the job's done. The less time that nitrogen is lingering around the environment where it might be prone to some of those losses. So urea, very quickly to be absorbed, and here's amino acid absorption. On this treatment here, they applied this one here, the glutamate, and you can see the triangle here. Look how rapid the uptake was. I mean, it's literally within the first hour or two. So under optimized conditions, you can uptake that nitrogen within a few hours through the leaf. You put that same nitrogen on the soil, especially in a dry year, then that nitrogen is going to sit on the surface and it may not even solubilize, let alone wash into the soil, get to the roots. All of that's going to take time. Whereas with a foliar, you can get that nutrition into the leaf literally within a few hours. So I'm going to scoot over these last few slides and let's have a couple of questions. Just to acknowledge how you apply it is really important. You want good nozzles, good droplet size. You want to get good coverage, good penetration, good sticking onto that leaf. So just general rules apply there. Different crops are more or less reactive, responsive is the word I was looking for. Different crops are more or less responsive to foliar spray. So plants that generally have very thick and kind of tough leaves, very waxy leaves, generally a little lesser responsive. It's not to say that they don't respond. Let's use brassica as an example. You know, they do have that tendency to kind of beat, the water does bead on the leaves. So just use a bit of a spreader sticker, kind of a wetter sticker, and spread that, reduce that surface tension, spread that water out, that droplet out, get it to kind of spread and stick on the leaf, and brassicas can still respond. So you can, there's ways around this, but as a general rule, there are differences. And I would also just say that as a general rule, your younger leaves the tender, fresh new growth is also generally more responsive than some of the older, more matured leaves. And then the key message from this slide in terms of the environment is the humidity. The humidity and the temperature that couples with that is absolutely a key factor in driving the uptake. So we want high humid conditions and not too hot conditions. And that's because with high humidity, we have the stomata are open. We have more of the micropores, the cracks and crevices in the cuticle layer. These are more open. These are more porous under high humid conditions. And this is what helps the nutrition pass in through that leaf. So as a general rule, foliars are more effective in that earlier morning, in the later afternoon and evenings when the temperatures are a bit cooler and the humidity is higher. So that's your optimum time to apply those high humidity. And the magic number here is around about 70 odd percent. Above that, you have very, very good humidity. Very, very good uptake. But I would encourage everyone to have a quick Google of this if you're unsure. This is called delta T, where we are plotting humidity with temperature. And all you need to do is look up your current temperature and your current humidity and plot those two things, and you can see if you are anywhere in this green band, then this is a good window for foliar application. So just Google image delta T, you'll find plenty of these examples. And so you can see anywhere here in this range is going to be good for a foliar application. So I would highly recommend going off this, because if you apply a foliar at the wrong time, when it's hot and dry and the plants are closed and retracted and conserving their moisture inside, they're not going to be open and porous and responsive to that foliar application. So the right timing is really key. Okay, in summary, sorry I went a wee bit over time, but let's have a bit of extra time for some questions. Hopefully y'all can stick around for a few more questions. So in summary, plants can make use of many different forms of nitrogen. And as good agriculturalists, we need to manage them all. It's not just about the main fertilizer inorganic forms. It's also about the organic forms. It's also about your soil health practices, trying to optimize soil biology, increasing soil biology so we can optimize some of the biological supply through that rhizophagy cycle, for example. Different forms, different nitrogen forms work synergistically. There is an opportunity to use a bit of them all. Combine some of the organic forms with your inorganic forms. That will actually improve the metabolism of those inorganic forms when you include some organic forms as well. So use a bit of them all. Diversity is beneficial. Diversity is beneficial in terms of these nitrogen forms. Your carbon-based nitrogen, or the organic nitrogen, is more efficient for plant metabolism and utilization. Okay, that's that metabolic shortcutting discussion. Any of the organic and peptide and protein type forms have this embedded carbon, and that makes them metabolically efficient. Nitrogen is not an island. It does not work in isolation. It needs those other synergistic nutrients in those little red boxes that we looked at it needs those other nutrients in order to help it do what it does. Excess nitrogen or imbalanced nitrogen can also compromise plant health. So this leads to lanky growth, imbalanced growth. It can make the plant more prone to pest and disease if we push too much growth, not push too much nitrogen, and it is not balanced with some of those other nutrients like the trace minerals that help to ensure good protein synthesis and good quality. And in terms of foliar nitrogen, it is particularly relevant, the amino acid and urea forms. These go very well together, and they are the preferred forms for a foliar approach. They are rapidly absorbed, and that means less losses. We can use less, we can be more efficient and gain those efficiencies. So, okay, I think that's it. Thank you very much, everybody, and let's have a bit of a chat and answer some questions, hopefully.
Awesome. That was great, Joel. I do have a question for you. At QLF, we have a product we call Amino 15. It's a little bit of a combination of some amino acids, some molasses, and even some urea. We've played with that specifically on corn at a lot of different application timings. V4 to V6 has been a good one, and even a little later in the season, maybe like R1, going out with fungicide. If you were just getting started playing with some foliar nitrogen applications, is there a timing that would have a higher effect on your nitrogen use efficiency?
Okay. Yeah, and that can depend a little bit on different crops. So there are some kind of optimized stages on different crops. So in the example of corn, I would say that yeah, I agree with you. Around about that V4 to 6 is a very good window for a nitrogen application. Another good window is around about V10 to 12. That's another good window right around there in terms of kind of high nitrogen demand and supporting that uptake at those key stages. And for where relevant, there can be another later one kind of around tasseling off when those tassels are kind of going brown, and we've kind of finished off a bit of a late season one there can also help to boost protein. That would be some of the key windows for maize. But I would say on your soys, actually kind of even on soybeans is really a good opportunity here, kind of around post flowering, around pod set. So a late season application is more relevant for, say, legumes. It can really boost the protein content of those beans. So the later one is more appropriate for legumes. And then on things like wheat, generally good and other cereals, generally good times are around about tillering, right on ahead of flag leaf, or kind of right onto that flag leaf. And again, if you want protein, kind of post flowering application, that late season one can also help to improve protein if that's also what you're chasing.
Awesome. All right, we have a question from the chat here. How long can you feed a corn plant with foliar nitrogen, and how much nitrogen can you apply at once? And I think this question kind of ties into another question that I had, just about how foliar nitrogen can affect your overall nitrogen rate. Does that make sense, Joel, or was that too many questions in one?
No, you just might need to remind me of the second. I'll probably forget it once I get rambling. So yeah, this is a great question. How much nitrogen can a plant take up, per application through the leaf? And of course, the obvious answer to that question is, are we talking about a plant that's this big, or are we talking about a plant that's this big? And how much leaf area do we have? So of course, a much larger plant with much bigger leaf area is going to be able to take up a lot more nitrogen per pass. So there's a scale there as we work through developmental stages. I would say that then, however, that said, there does start to be an upper end, which maybe is more important than those differences in terms of, say, nitrogen scorch or burn or application and that kind of thing. So just to give you then a guide, I would say that most farmers that I'm familiar with around the world, on a range of different crops here, they would typically be applying somewhere in the range of 10 to 15 pounds of actual nitrogen per application, generally in that range. And I would say, but again, there might be subtle differences between different forms and the salt index. So, for example, look, UAN, your 32 solution has a higher salt index than, say, urea. So there can be other scorch factors from salt index here. But just as a general guide, I would say it's in that kind of 10 to 15 pounds. But that said, I do know that a handful of examples of folks going higher than that, creeping up to as much as even 20 pounds, say, in a single application. I've seen that also on wheat. So look, I would encourage, if you're new to it and or experimenting, look, I would safely say around that 10 is a really safe starting point, and then I would kind of experiment and test based on your crop, your variety, your water quality. There's many other factors here that are going to influence the potential kind of factor there. But in that 10 to 15, I'd say is generally a pretty good general answer of what most farms seem to be applying. And to your second question there, Shannon, can we use foliars to kind of partially substitute our soil-based strategies? And yes, this is the kind of part of the benefit or the goal of the foliar is that when you deliver foliar straight into the leaf and you bypass the soil and therefore you bypass the losses. And remember, I mentioned particularly with nitrogen, which is one of the worst, where we often have only around about 50-odd% nitrogen use efficiencies, nitrogen uptake use efficiencies. There's the answer. There's your opportunity. Often you're losing a lot of the nitrogen that you apply. Now, don't get me wrong. That's not to say that the other 50% is all lost. I mean, some of that other 50% can be held in the soil organic matter, held in the soil biology. So some of that nitrogen you're still holding in the soil environment, but definitely some of it you are also losing. That's the fraction that we really want to try and capture and reclaim. And if you can foliar apply and get that nitrogen directly into the leaf, that fraction there is the fraction that you can start to cut back on. So practically speaking, what that means is that farmers that I'm familiar with using this foliar approach, yes, they typically do, and again, this is a gradual process, but they do generally begin to reduce their starter nitrogen applications. So they'll start to wind those down and use less up front, and then you're topping up later on through the foliar through that more efficient means. So I would find that particularly in an annual cropping plant also, you do still need a bit of upfront fertility in the soil. And that's just for that obvious reason is that before you can start foliar feeding, you need to grow a canopy. So you're still going to need some upfront fertility. You've got to grow that leaf area first, grow that canopy first. And once you've got, good, decent canopy closure and enough leaf, for example, well, now you can kind of finish that off with the foliar approach. So generally, I would encourage farmers to kind of maybe experiment with this and start slow. But yes, there is that opportunity to use less throughout the season because you're losing less. And when you lose less, you can use less. And that's that key point there. So yes, the foliars, because of that efficiency gain, not only in terms of targeted application straight into the leaf and therefore reducing losses, but also in terms of having more control over what form of nitrogen gets into the plant. So that's that nitrogen utilization efficiency. Then through the foliar, you can ensure that the urea you apply is getting into the plant as urea. You can ensure that the amino acids that you're applying are getting into the plant as amino acids. And those metabolically efficient forms are what's going to help you also use less because of that greater control of what form gets into the leaf.
Awesome. Well, I am not seeing any other questions at the moment. Big thank you to Joel for joining us from so far away today. Like he had mentioned, if you Google Joel Williams soil, his website will pop up, which is integratedsoils.com. But like I said earlier, and he mentioned it a couple times, Joel also has a foliar nitrogen course on his website that is an awesome course. So if this little lecture sparked your imagination, go ahead and get on there. He also has lots of YouTube videos, and on his website, you can see some of his other speaking engagements. So feel free to go check that out. But a big thank you to Joel for joining us today. And helping us believe in what we're doing, thinking we're on the right track, and doing a lot to, further this nitrogen conversation. So big appreciation to Joel. And if anybody has any other questions, feel free to reach out to me or Maddie or your other RSAs that you know, and we will do our best to get them answered. So thank you so much for jumping on.
Pleasure. Thanks very much for having me. Yeah, all the best to everyone for the rest of the growing season.
Thanks, Joel. You too.
Cheers.